presentation is based on an ongoing project on molecular analysis in forensic science. This is conducted in the Department of Biological Science here at Kean University. This project explores the use of portable nanopore sequencing technology for the extraction and sequencing of DNA from skeletal material and insects, two types of biological evidence that are often encountered in forensic investigations. Recovering DNA from degraded biological material remains one of the most persistent challenges in forensic science. In many forensic cases, DNA can become fragmented and difficult to analyze using traditional laboratory methods, especially in cases involving environmental exposure, fire damage, or advanced decomposition. Additionally, another common drawback of DNA analysis could be the delays caused by the laboratory processing as well as the high cost of them. To address these challenges, this project aims to evaluate the use of the Oxford Nanopore Minion MK1D, a small portable sequencing device capable of generating DNA sequence data in real time. The Minion is approximately the size of a USB device, on the contrary of the traditional sequencing platforms that require large laboratory infrastructure. Therefore, it can operate using portable equipment, allowing it to be deployed outside of a traditional laboratory environment. DNA extraction and sequencing in this project will be performed using a commercially available kit designed specifically for nanopore workflows. The goal is to develop practical protocols for recovering and sequencing DNA, both from skeletal remains and from insects associated with decomposition, such as those found on human remains in forensic contexts. Nanopore sequencing works by passing individual DNA molecules through tiny biological pores, nanopores, embedded in a membrane. As the DNA strands move through the pore, changes in electrical current are detected and interpreted to determine the sequence of the nucleotides. One important advantage of this technology is that it can read entire DNA fragments from short segments to extremely long strands in real time. Another advantage of the Minion is that it allows for researchers to use the device in field settings, is the built-in temperature control, allowing therefore to operate under ambient environmental conditions. Researchers have even described portable nanopore sequencing systems as lab in a suitcase, highlighting the potential for the use in remote or resource-limited environments. Whereas nanopore sequencing has already been used and has shown strong potential in clinical diagnostics and environmental genomics, its systematic application in forensic context remains relatively limited, especially in cases with degraded samples. This project aims to assess whether portable nanopore sequencing could support rapid genetic analysis directly at forensic scenes or during early investigative stages, and to assess that material that is commonly found in crime scenes and give usually relatively degraded samples will be examined. Thank you very much.